Okay, hello. Welcome to Graham Rounds Neurology. Graham Rounds. I am very excited today that we have Dr. Roger Simon with us from Morehouse School of Medicine in Georgia. Roger is outstanding neuroscientist, somebody who really I saw speak, I guess, for the first time about 11 years ago at an ANA meeting and talking about ischemic preconditioning, and I was so intrigued by what he had to say that I decided that's where I wanted to work in my research. So he's an inspiring speaker, and I hope you guys will all enjoy that. He's done a lot of work on genomics and stroke and genomics and ischemic preconditioning. He trained residency at University of California, San Francisco, was at UCSF for many years. He is the author of many research articles, and maybe notably for the residents, he is a co-author on the Lang neurology textbook, which I think probably all of you have probably at least seen at one point. Besides his research career, he's an outstanding clinician. And today, he's going to be speaking to us about his latest work in the genomics of stroke. Thank you.
It's great to be here. The Pacific Northwest, there's just nothing like Puget Sound, I think, to deal with whatever problems you have with your soul at the moment. Thanks for mentioning the textbook. We put a lot of work into that textbook. It took 10 years to write. It took 1 year to proofread for the first edition, and I have it sitting on the table in my clinic room every clinic session that I do, and where I used to say to patients, "Oh, just a minute, I'll be right back," and I would go in another room and look something up. I don't bother anymore. I say, "See this? I wrote it." So now I can look in here and find out what muscle I want to test. Okay, so I'm going to talk about stroke, and so in 30 years we've actually learned an awful lot about stroke. We went back to Jim Brierly's original experiments from the 40s and saw the earliest morphologic changes in an ischemic neuron seen here with toluidine blue staining the microvascularization of the cytoplasm. And Brierly said in the 40s that if we knew what this was and if we knew what caused it, maybe we could come up with a therapy for stroke. And then with Brian Meldrum, we looked at calcium in ischemic brain here in the control specimen. The calcium pyroantimonate staining is in the presynaptic vesicles here at the glutamatergic synapses about to be released, and here it is in an ischemic neuron and at a different magnification. All of these little dots of pepper are intracellular calcium accumulation, and and what these vacuoles are is mitochondria. So there's the organelle that is the first to be affected by intracellular calcium toxicity. And Boussachew gets credit for the calcium hypothesis in ischemic pathophysiology, but it was actually Brian Meldrum's. Anybody that wants to buy me a beer, I'll tell you the story about how that happened. And we can attenuate calcium flux in the brain, first by glutamate antagonists, but more recently by calcium sensing ion channel antagonists. And we can have a time window that's translational. Here's 1 hour, 2 hours, 3 hours, and 5 hours later by using a drug to block these channels to prevent calcium toxicity and attenuate stroke injury. So we've gone a long way in regard to stroke pathophysiology and therapeutics, but there's a problem. And the problem is that this has been done over 1,000 times and nothing's worked in humans. And so, after spending several decades working through my own assortment of individual drug pharmacology for stroke, about 20 years ago, just in a moment of desperation, I did this experiment. And the experiment has to do with an antibody that we had from a group across the street was studying something in the lung, which I can't even remember what it was. And it's a very, very clean antibody for heat shock protein. In other words, it's an antibody that stains a new protein that the brain has made. In other words, the brain responded to an event, to a stress, in this case, hemispheric ischemia, and it did something about it. And it made a new protein. And you can dial this up and dial it down, and so we dialed it to the point that the brain would respond by making a new protein. It would respond, but it wouldn't kill the cells. So here we had a brain that saw what the problem was, and it did something about it. And then we took brains that had this stress response, and we gave them a stroke. And just to ask the question, has the brain learned anything over the millennia when they were dealing with saber-toothed tiger bites or something to the cortex? Have they learned anything in regard to how to deal with this problem that's better than what Roger was doing with trying this drug, this, the next drug, and the next drug? And the answer is yes. And this was kind of surprising that here's a stroke in the volume of stroke, the area under the curve, and this is an identical stroke in an animal that had seen the problem before, and they had seen a little bit of ischemia, enough for them to have to, for the brain to have to do something, at least make heat shock protein, but probably because that's the antibody that we had, but undoubtedly it did other things as well. And the brain was capable of attenuating the response to and exactly the same duration of ischemia. Actually, these are chronic occlusions of the middle cerebral artery. And then we played around with this for a number of years to get the timing right and to be able to do it in a mouse. And so here is the suture occlusion technique that Phil Weinstein worked out at the VA hospital in San Francisco. 60 minutes of transient middle cerebral artery occlusion by an intraluminal suture, and you get this big an area of ischemia. And this brain had 15 minutes of ischemia prior to an hour of ischemia. So that was exciting. That was exciting. So it knew what it was getting into, and now when it sees 60 minutes of ischemia, it's only this end artery distribution in the lenticular striate arteries that infarcts, and the entirety of the cortex is spared. And this is a 70% reduction in infarction, so that the brain has this enormous capability of seeing exactly the same amount of ischemia that would produce a big stroke and doing something to attenuate the area that's injured by what would be a devastating amount of ischemic injury. And there may be some correlation in humans. There's about 6 of these studies now, and they didn't all show this, but the plurality shows this, and that is that if you have a TIA, you're more likely to have a mild stroke than you are if you didn't have a TIA prior to your stroke. So that there may be a preconditioning phenomenon that occurs in humans as well. And I think I'll skip this. The point is that the cardiologists love to take responsibility for having discovered the ischemic tolerance phenomenon, but here they are on my list, and there were lots of things in the nervous system prior to that. And then the one that I really, so here's Kitagawa's famous experiment with global ischemia and neuronal injury, where he showed ischemic tolerance in the gerbil. Here's our experiments in stroke, where we show white matter and gray matter protection. And then there's this experiment that I think is really cool, and that is that if you are subjected to, as teenagers are these days, very loud sounds, you can get injury to the cochlea and the hair cells in the inner ear. And if you give a little bit of loud sound exposure, then it attenuates the development of injury to those hair cells with a prolonged, very loud sound exposure. And they called this toughening. So this is resistance to injury in the ear. So what causes, what machinery in the brain causes this endogenous neuroprotective phenomenon that's so potent? So we thought about this, and we thought about, well, there's some neuroprotective proteins in the brain like BCL2 and BCLX, at least the long form, and the Pak-Chan's experiments with AKT. And then there's some bad guys too, like BAX and BID, and opening of the transition pore in the mitochondria, cytochrome C. The caspases, caspase 9, caspase 3 are bad guys. And so we started kind of walking down the line of doing these experiments. And you know what? Before you even started the experiment, you knew what the answer was, because you knew who the good guys were and who the bad guys were. And so it wasn't a very interesting time in the lab, except learning these pathways, I suppose, was of some interest. And then something happened, and that is that the mouse genome was cloned, and the Affymetrix company developed the microarray technique for looking genome-wide at changes in a given setting. And this was just when I went to the Robert Stone Dow labs in Portland, and there was a woman at OHSU, Chris Harrington, who was a senior scientist at Affymetrix, and she had just moved to the medical school in Portland and set up a microarray laboratory. So I said, wow. So here's the chance to really find out what is going on in the in this brain that's endogenously neuroprotected, that we can look at the entire genome at a moment in time. And so we set up the experiment, and we preconditioned. We had worked this out. We preconditioned mice with 15 minutes of suture occlusion. And then we used 60 minutes of suture occlusion, which was enough to produce a stroke. And we looked with the Affymetrix microarray chips to see how many genes were regulated. Now, don't ask me about the statistics here, because you can dial this up and you can dial it down, and when you're looking at 25,000 data points, there's a lot of potential for changing the system. But what we saw was that the number of genes regulated in the brief preconditioning ischemic stimulus, this is suture occlusion of the middle cerebral artery, and the number of genes that were regulated in prolonged ischemia, enough to cause a stroke, using the same technique, were completely different. And then we took the preconditioned brains and we challenged them with prolonged ischemia, and we saw, again, a different set of genes, and when you put it all together, there was no overlap between the genes that were regulated in preconditioning, even though it's spelled wrong, and the genes that were regulated in stroke, and the genes that were regulated in tolerance. There was no overlap. And so the point here is that the brain is able to reprogram its response to ischemia, that you can take the same amount of ischemia, and if you regulate these genes rather than these genes, you can get a protected readout and not an injury readout. That preconditioning reprograms the response to an ischemic challenge. That it's the same ischemia, it's the same duration of ischemia, it's the same brain. I won't go into this, but there's no cerebral blood flow piece to this puzzle. The brain is able to do this reprogramming. And then we found something that was completely unexpected. And when you do an experiment and you get something that's completely unexpected, this is really like the most interesting thing that can happen to you. I mean, I knew what was going to happen in this genome array package here. All the good guys were going to be turned on, and I knew who they were, and all the bad guys were going to be turned on, or were going to be turned off. So we were going to see all the BCL2 good guys were going to be upregulated, all the BCL2 bad guys were going to be downregulated, all the heat shock proteins were going to be up, all the caspases were going to be down, and that's not what we saw. We saw widespread suppression of gene transcription throughout the brain, and a divergent response to the gene ontology characterization of the regulated genes. So its turn off is the phenomenon of neuroprotection. It's the reprogramming response. And we, just to give a little flavor to the reviewers of this article, we said, and it's just like happens in hibernation. And this was, and there is some literature to support this. Kelly Drew works on hibernating ground squirrels, and John Hollenbeck as well. But I don't think it's a simple refrigeration point of view, but maybe that catchy title helped get it published. I don't know. So transcriptional suppression is the good actor here in the protected brain. But there's very limited overlap of how the brain responds to different scenarios of ischemia to produce this reprogramming. But it's a bit of a paradox, because if transcription precedes translation, then this shutoff of the biology of DNA, RNA, protein kind of goes against a known fact, and that is that the tolerance phenomenon is protein synthesis dependent. So this didn't quite make sense. But the widespread effectors that are involved in this process suggested to us that there may be some epigenetic components, since there's so many pieces of this puzzle involved. And so we needed to do proteomics, because this is how life works, is it? And I said in a conversation a couple hours ago, well, in regard to these areas of truth, maybe we have to check with Donald Trump. But I think this is the way the life works. And so we wanted to look for protein effectors. And now, as I go through this talk, I have a tendency to use the word we, and we all do this. And I just wanted to point out quite clearly that when I say we, I never mean me. And so the we here in regard to proteomics is An Zhao, who's a protein chemist that's been with us in Portland and in Atlanta, and she's just a whiz. And Martha Staples, who's a chemist who was at Oregon State University and then took a job with the Waters Corporation, who makes the mass specs. And so this is really their work. And so we did exactly the same experiment. We looked at a little bit of ischemia that produces preconditioning in the brain, and we saw what proteins were regulated, and then we looked at the proteins that were regulated in brain that had enough ischemia that was going to cause a stroke, and we saw a lot more overlap than we saw at the RNA level, but there were still proteins that were uniquely altered. And then we did the third experiment, and that is to take preconditioned brain and challenge them with a prolonged piece of timing of ischemia. And so here's what we got. There is a lot of overlap at the protein level, but there's still a group of proteins that are only found in preconditioned brains that are then subject to prolonged ischemia. So it's these proteins that we were particularly interested in. The upregulation and downregulation doesn't look like the data at the RNA level. There was as much upregulation as there was downregulation. And then when we looked at the gene ontology data, we saw that chromatin and DNA were areas that were particularly found in these protein group that was uniquely upregulated in the preconditioned and tolerant brain. So we hypothesized that modulation of these chromatin remodeling proteins is what drove the suppression, that these would be, we hypothesized these would be epigenetic mediators that would be responsible for the decrease in transcription that we had previously reported. So then if you look at these groups of proteins that come out of a mass spec, and these experiments are, I don't have to tell you, they're way more complicated than the gene chip microarray experiment, and this isn't anywhere close looking at the proteome, and there's no way to really do that. But in the group of proteins that were found to be upregulated only in tolerance, we saw two that got our attention, and it was histone 2A and Polycomb protein sex comb at mid-hind limb homolog 1 gene. And because these genes are involved in limiting DNA accessibility, and I'll show you a compelling picture of that in a minute, they're part of the Polycomb repressive complex that maintains a transcriptionally repressive state, and this particular group of Polycomb proteins regulates transcription. It's a gene silencer, and it has transcriptional repressor activity. So what we discovered was a group of genes that were epigenetic transcriptional repressors. Again, we is An Zhao. And so what about these Polycomb proteins? The story is kind of interesting. They were discovered in 1947 by Pamela Lewis, and she was looking at male fruit flies. And male fruit flies have sex combs on their forelimbs that are used in grasping the females in mating. But what she found was there were mutants that had sex combs not just on the forelimbs, but on the, you remember the insects have 6 legs, on the mid hind limb and the hind limb as well. And so she termed these mutants polycombs. They had multiple sex combs. And then Ed Lewis, some relation, worked on this for the next good deal of his life, and in 1995 won the Nobel Prize in Physiology by showing what the protein effectors of the Polycomb phenomenon was, and he called the proteins Polycomb proteins, and they shut down, they're gene silencers that shut down developmental genes in the mid segment and the posterior segment, and do it permanently. And Gidey at WashU has recently shown that using hypoxic tolerance, that the next generation retains the tolerance neuroprotection. Well, there are only 15 of these genes in Drosophila, but in mammals there are many hundreds, and they form multi-protein complexes, so it's much more complicated in humans. And I'll show you a little cartoon in regard to how these work, but the Polycomb repressor complex 2 trimethylates histone H3 at lysine 27, and it does so in about 1,000 places on the chromatin, and it produces gene silencing. And I'll show you that cartoon. So here's a picture of the sex combs, and I just couldn't resist making the point about why I was so familiar with this stuff. And the answer is that I have a master's degree in science, and it happens to be in entomology. And so I was able to bring this to good use in this neurobiology exercise. So here's just a couple of experiments that we did to try to prove the point that this group of Polycomb proteins are the effectors of the tolerance phenomenon. So here are 3 of the Polycomb repressor complex 1 proteins, and the thing to look at here is just the injury column. So here are the 3 Polycomb proteins that we're interested in. Here are the injury column, and here's the tolerance column. And I think what you can see is here, this is sham operated, this is tolerance, that there is an increase in the tolerant cells, but a decrease in the expression of these proteins in the injury column. So that in tolerance there's a upregulation of these proteins, and in injury there is a downregulation of the Polycomb group proteins. And then to do the standard experiment, if you knock down these here are 2 Polycomb proteins, you attenuate the phenomenon of tolerance, and if you overexpress them with a viral vector, then you attenuate the degree of injury in these cell culture systems. And if you do it in vivo, here's the amount of a measure of infarct volume. Here's the tolerance phenomenon at the 15 minutes of preconditioning ischemia before 60 minutes of prolonged ischemia. And then here's the siRNA for these 2 Polycomb proteins, and this is a scrambled control. And what you see is that if you do the tolerance phenomenon, the preconditioning and the permanent and the 60 minutes of prolonged ischemia, if you knock down either of these Polycomb proteins, you attenuate the degree of neuroprotection that the tolerance phenomenon gives you. So how does this system work? So this is a little cartoon. So there's Polycomb repressive complex 2, marks. It puts these trimethylation marks at the lysine 27 residue of histone 3, and they stay there permanently. And then Polycomb repressive complex 1, this group of proteins, recognizes these marks, and it binds to these sites on the chromatin and attenuates gene transcription there. And here's a picture of a normal piece of chromatin, and now one exposed to Polycomb repressor complex 1, and you can see it really does tighten up the chromatin with that exposure. So this is what we think is happening in the preconditioned brain. We think that the Polycomb proteins are producing broad repression. They bind to hundreds to thousands of genes on the chromatin, and that's the proteomic signature of tolerance. But it can't be that easy, can it? And it's not, because it turns out that there is an opposing group of proteins, and they're called the trithorax proteins, and they are also epigenetically effective, and they do exactly the opposite, and that is they activate transcription. And so you can see where, and they produce this type of a balance. And now you can see where what we want to do makes sense, and that is we want pro-life. We want to do silencing and protect the brain during a stroke. So we want upregulation of Polycomb proteins. We want downregulation of trithorax proteins, which I'll show you an image of in a second. But this isn't always good, because if you that's great if you're having a stroke, but it's not great if you have a glioblastoma multiforme, because then you don't want this pro-life situation to just go on and on and on, because you develop oncogenesis. And it turns out that this whole trithorax story is well known to experimental oncologists. And they're interested in doing exactly the opposite that we are, and they're trying to knock out the Polycomb proteins, or upregulate the trithorax proteins. And here's the same cartoon, but I wanted to show you this picture. So here again in cell culture, here's injury, and here's Polycomb proteins are downregulated, but in tolerance they're upregulated, and the trithorax proteins are the opposite. In injury they're up, and in tolerance they're down. So it's this seesaw puzzle. So this is what we think. We think that there's a genomic proteomic view of ischemic tolerance, that preconditioning reprograms the genomic response to ischemia, that it's right there in the brain's biology, the ability to take 100% infarct and reduce it by 70% to a small infarct. And we think that gene transcriptional downregulation is a central feature of this reprogram response. And we think that protein synthesis and tolerance, there's differential protein expression of epigenetic regulators, Polycomb and trithorax protein. And the bad news is that they regulate hundreds to thousands of genes. And so maybe this is the reason that none of the single drugs that were brought forward in that paper that reviewed over 1,000 neuroprotective compounds worked. But endogenous neuroprotection has huge therapeutic potential by reprogramming the genomic response to brain to induce tolerance. That this machinery is in the brain, and it's highly effective. Okay, so as was pointed out in the introduction, I went to a place that actually I didn't know about, 5 or 6 years ago, Morehouse School of Medicine in Atlanta, and I went there because an old colleague from UCSF, Zach Hall, who was also the director of the NINDS during the Clinton administration, ran into me at a meeting in Fairbanks, Alaska in February at 50 below. And so we couldn't do much except sit around and talk about where we had been and what we had done and so on. And I said, look, I'll just send you a copy of my CV, and you'll see what we've been up to since I left UCSF. And about 2 weeks later, I got an email from him and said, there is a job in Atlanta that has your name written on it. That's always kind of an interesting phenomenon. And so I went there to look at this place. And basically, they wanted somebody to set up a translational program in experimental stroke that specifically looked at stroke in African Americans. And I knew from 20 years at San Francisco General Hospital that stroke looked different in African Americans, and I was kind of curious about it. So I thought that maybe I would give it a try. And having made that decision, actually, and our neurophysiologist and our genomics guy, they said, we want to go too. And so we all moved to Atlanta. And I thought that one thing that I could do there was change my animal model. That I've been stuck with rodents for a long time, and here at the Grady Memorial Hospital, where the clinical service for the Morehouse Medical School is 90% African Americans, and so I could have a new animal model. I could use humans as my animal model. And as a number of us were talking about an hour or two ago, one of the most exciting things that has happened in the clinical stroke arena is Frank Sharp's crazy idea that you could take blood cells out of peripheral blood and look to see what genes they were expressing and make some extrapolation to what was happening in the brain. Because after all, these blood cells were swishing around in the brain just a little while ago. So we thought we would look at that in African Americans, because Frank, I think, had 7 or 8 African Americans, and stroke is different in African Americans. And we published a paper a few years ago showing that in TPA, the response of African Americans to TPA is quite different, particularly in women who have virtually no response to TPA. Maybe some of you saw me on Fox News at 8 o'clock in the morning a few weeks ago talking about this. So, but we thought we would do better than Frank, and Frank is like one of the greatest guys in the business, but he was using microarrays, and we decided to use next generation sequencing so that we could sequence all the pieces of RNA. And again, we is not Roger. It's Rob Meller, who's an Oxford UK trained molecular pharmacologist. So we can look at the protein coding regions as the Affymetrix chips do, but we can also look at the intron regions or the intergenetic regions, and we can get at least 3 times the data and novel data that can't be gotten from the gene chip microarrays. And so here are a couple of pieces of data that are impressed. The first confirms with a different technique with gene sequencing what Frank has discovered, and that is that you can make a diagnosis of stroke with a sensitivity of, in our data, 88%, at a specificity of 100% by looking at these hierarchical clusters of gene expression. So here are the hypertensive control patients from our outpatient clinic, all hypertensive African Americans, and here is the patients with an MCA occlusion, again, hypertensive African Americans, and it's not hard to see the difference. And you can tell the degree of the injury using a mathematical three-dimensional technique that you can read about if you go to Wikipedia on Google, and you can separate them into, based on NIH stroke scale, minor, moderate, or severe strokes, and look at their mathematical characteristics. And here's what Frank showed, and we would confirm that TIAs are intracerebral hemorrhage or atherosclerotic or cardioembolic stroke or small vessel stroke all have specific transcriptomic signatures in the blood. Here, in our situation, they're in African Americans. And then something Frank didn't do.
Can I ask you, Roger,
Yeah.
what the time delay is? When are you making these measurements of blood?
Yeah, it's within the first day after the stroke. So we take the blood draws at 8 o'clock in the morning at anyone who had a stroke who was admitted the day before, but they had to have had the stroke the day before. And then here's the most exciting part of this IMPRESS data. First, you can see from these transcriptomic patterns that are taken on admission in the emergency room, who has a minor stroke, a moderate stroke, and a severe stroke, so that the degree of severity of the stroke is differentiated. But here is something that's actually fairly spectacular, I think. Remember, this blood test is taken the day the person came in, within 24 hours of their stroke. And then they're in the hospital for some period of time before they either leave the hospital or are transferred to a chronic care facility. And this admission transcriptomic signature, the one that happened in the first 24 hours after after the stroke, predicts with a remarkable degree of certainty who will have a good prognosis and walk out of the hospital, and who will have a bad prognosis at the end of their hospital stay and not improve. We drew the line at 60%. You had to get a 60% improvement.
How fast did these genetic changes occur?
Within the first 24 hours. I don't know if it's in the first hour or 2 hours or 3 hours, but it's within. Once these genomic changes occur, and I think it's 1 hour, 2 hours, 3 hours. There's not enough data to say, but 6 hours to 24 hours, it doesn't change. So it's pretty fast, and it stays there for some time. So I think this is kind of amazing that you can see a prognostic signature that is already determined by the biology in the brain. When you have a stroke, what your degree of recovery is going to be. So we are involved in trying to crank this up and look at other nervous system disorders. So I think this is the future. I think it's transcriptomics. I think it's molecular medicine. I think that this is what, when our kids get sick, this is what will be the diagnostic test. And nobody knows how to use a stethoscope anymore anyway, at least in my experience in the hospital. This is personalized medicine. This is your genomic response. This is your transcriptome. This is precision medicine. What did I write here? Precision medicine. Sorry. And that's why that famous Lang textbook, my section, I dictated it. I didn't write it because if I had written it, there would be all these kinds of words in there. And the future is the diagnosis and prognosis of stroke and maybe other CNS disorders. So we're just starting to look at epileptic patients. We have post-concussive patients who have their own sequence that you can tell if they've had one concussion, two concussions, or three concussions by these transcriptome profiles. And then the people who were the genomic stuff, Chris Harrington is the person from Affymetrix. Mary Stutzelpour was the faculty person at OHSU who was involved in the studies, and our group, the Proteomics, were An Zhao and Martha Staples. The Transcriptomics are all Rob Mellers. And then there are two areas that I didn't talk about, and I'm not going to, the microRNA stuff and the single-cell electrophysiology. So that's it, folks. That's stroke in 2016, at least from my viewpoint. Thanks for the invitation.
So the genomic or transcriptomic profile that was indicated a good prognosis, were you able to correlate patients that have that profile with a history of TIA? I mean, kind of going back to a preconditioning.
So can we, do we know what the transcriptomic profiles are in TIA patients? Are they predictive of tolerance? Well, no, because the patients were mice. With exception of those last two slides I showed you, all this work is rodent work.
Right, but in the patients that you did those blood analyses on, was it more likely to see that good prognostic pattern on the initial blood test in individuals that had a history of TIA?
If you just had a TIA, could you see a transcriptomic pattern? We don't have enough data to do that because we're subject to who the emergency room chooses to admit, and they usually don't admit TIA patients.
But this could be history, Roger. In other words, those people that were in the good prognostic category, is it possible that when you took their history, you discovered all of those people had a TIA within the last 2 months?
Yeah, is there some history of a TIA?
Right.
We don't know. We're stuck with the fact that all this stuff comes out of the emergency room. That's a totally reasonable suggestion. But if we saw the degree of good outcome in more than half of the patients, I don't think more than half of the patients had TIAs. I wouldn't think.
In your analysis of the differential gene expression with good and bad outcomes, in your paper, I guess you parsed out what general families of genes are involved in the protection versus.
Yeah. What families?
What cell type is it?
Oh, what cell type? What cell type is involved in the production of the genes that are neuroprotective? These were mainly in neurons, but that's all I can say.
How long does the tolerance last? And
Yeah, how long does tolerance last? That's really important, and it gets to this TIA question too. Again, I can only tell you in rodents. In rodents, some people have reported, we don't think this happens, that there's tolerance in the first 24 hours. We think there isn't. But there is tolerance at 48 hours. It's maximum at 72 hours, and it goes away by 7 days. So that's it, in a rodent.
So in the blood samples that you use, do you know what percentage of the RNA that you're looking at is coming from erythrocytes and what percentage is coming from leukocytes or exosomes? Do you have any clues from the bioinformatic analysis of those genes as to whether or not the big shifts are coming from one particular cell type or other within the blood?
Which cell types are producing these transcriptomic changes? We've just used whole blood, so it's everything that's in the blood. The problem is that when you start to pull out these individual cells, as Bard has done, for example, in a well-recognized paper in Circulation, when you begin to play with these cells, they change their gene expression because you either are squishing them or pulling on them, and these mechanical forces change gene expression. So we have, as Frank Sharp has, just decided to just use whole blood.
But bioinformatically, you can look at certain messages which are restricted to specific cell types, and that should give you some information as to where the major changes are coming from.
So could you do this in a bioinformatics way in regard to knowing that certain genes are likely to come from certain cell types? I guess you could do that, but we haven't done that. That's a good question.
So, Roger, that was very interesting and provocative. I want to back up to the first part of your talk where you were doing the animal experiments and showing us the various Venn diagrams. You didn't talk at all about the changes that you get with preconditioning. So in some ways, I'm of the school that the stuff that comes later may or may not be important. But what's really important are the changes that you precondition. What are those? I mean, there's those things have to be responsible for the benefits that you get when you do the challenge.
Great question. The question comes down to preconditioning is the whole thing. It's not tolerance anyway. It's preconditioning because that's what sets the brain up. Julie Sextad in the lab did an experiment looking at preconditioning, and her paper suggests that it's microRNAs that set this up. And I haven't followed up on that, but if you use her name, Lasardi is the first author of that paper. But you're dead on. Preconditioning is the thing.
We have someone here who's asking you a provocative question. If you can read it. That would be our own David Terschwell.
Prognosis is largely driven by
Yeah, good question. So this guy is a clinician, and he's very good at knowing who's going to make a good recovery and who's going to make a bad recovery. And he wants to know if this has been controlled for
He appreciates those assumptions about who it is.
Yeah. And that is a very important question, and the answer is we haven't. And the huge number of patients that were in both categories, and the fact that the distinction between these two transcriptomic analyses gave 100% credence. We went with it at least as an opening offering, but the types of analysis that the questioner suggests are critically important and will be done.
In your human subjects, do you see the same thing with large vessel territorial stroke as opposed to microvascular stroke lagoons?
Well, we can. So, is there a difference in the various stroke types? Yes. The readout is different in each of the stroke types, and maybe the most eye-opening piece of data we found, but Frank Sharp was the first one to show it, was that if you have now I'm making this up a little bit, but this is the point. If you have a middle cerebral artery occlusion caused by in situ sclerotic occlusion of that blood vessel, you get a facet arm greater than leg stroke on the other side, right? But if you have a cardioembolic stroke that occludes exactly the same blood vessel with exactly the same physical phenotype, the patients look the same, but their transcriptomic profile are different. You can tell which group of patients had a cardiogenic embolus causing this occlusion and which group of patients did not. And he and Frank has a theory that most of the cryptogenic strokes are probably cardioembolic based on this transcriptomic analysis.
So in your human subject cases, have you looked at the RNA expression level as compared to your animal model challenge and preconditioned cases for similarities in signatures?
Yeah, so
that's a very specific type of stroke, and you're actually
right.
able to.
So what's the similarity or difference in the transcriptomic or proteomic, for that matter, readout in the animals compared to the humans? So it's something that's a little bit scary, and that is that, and I've talked to Frank Sharp about this too, and it's his experience. There is very little overlap between what we see in the rodents and what we see in the humans, and that's kind of frightening for all of us that have spent years and still do working on rodent models of these diseases.
Roger, I'm just trying to think about what's happening to these blood cells that you're measuring after a stroke that would make them have a different transchromic profile. And one trivial thought is that if you have a worse stroke, you would have more red blood cells that would have seen a hypoxic period in their transit loop.
Mm-hmm.
You could test this easily enough by exposing red blood cells to variable degrees of hypoxia, maybe taking mixing them in different proportions to look at gene expression. Have you thought about this? I'm sure.
Well, so can you use hypoxia to stimulate red blood cells and see what their transcriptome is? Well, of course, that's what Gaudet does at WashU. That's his stimulus for producing tolerance. We haven't done that. We've tried to eliminate hemoglobin because it's such a big component of gene expression in whole blood samples, so we haven't done that.
But what I'm really asking is, why do you think that red blood cells change?
Oh, I think that these are immune modulatory cells. I think they're white blood cells, and I think that they're seeing new proteins. I mean, it's a self versus not self. I mean, it's not any different than if you had bacterial meningitis. I think these are immune surveillance cells that are seeing something unusual, and they're making a protein. And to give another negative piece of this biology is that therefore I think that we may not have any biological effector data from these genes. We see pattern recognition information on these heat maps, but I don't think we're seeing biological expression. I don't think the answer to preconditioning or tolerance is in this gene array.
But Roger, if that's the case, and these genomic pathways that you and others have identified have pointed out specific molecular pathways in the immune system, and when you genetically delete those pathways, you can abrogate ischemic preconditioning almost entirely. So I don't understand this idea that we're looking at an epigenetic phenomenon if the genomics have in fact predicted pathways that can abolish the phenomenon.
Yeah, so I'm supposed to repeat the question. This question is too complicated to repeat. It's basically there are already pieces of cell signaling biology that have been discovered that will block the tolerance phenomenon. So it's not all that as murky as I've said it was. And all I can offer is there's data on both sides of the street, and we'll just have to see how it comes out. All right. Thank you very much.